All right, everybody. Here's what's up. You're going to be making that foldable that you just constructed, hopefully. It's right here, right? Make sure you don't need to use color for the pictures. All right. The definitions for the first two are given to you. You'll see those on the next slide. But each should have the definition, a sentence using that word and showing that you understand what it means, as well as a little sketch to show what it means to you, helps remind you of that term. So that's number one. I estimate if you just do it, it's going to take you like 20 minutes. And that's if you're like putting in good effort, which I expect all of you to do. 25 minutes. All right, second thing is the cell cycle diagram. So this is on page 445, or you can use the Gizmo, which also has really great information and it's very clear and will give you exactly what you need to do this activity. You're going to use your brain to answer these two questions. Why is it important that cells' DNA is duplicated, copied before cell division? And what do cells need to do between divisions to make sure they don't just get smaller and smaller, which would totally suck? So use your brain, write your name. You're also going to be using the textbook, page 445, or Gizmo, to write what is interphase, mitosis, prophase, metaphase, and so on, in your own words as best you can. So there should be text underneath each of these labels, and these ones too. All right. And then you're going to draw in color. This one you use color. You're going to draw what's going on in the cell at that time. So you're using color. You'll see why, because there's like little pieces and it's just going to look like a mess if you do it all the same color. So please use colored pencils. They're on the ends of the lab tables. So that's that one. This I think will probably take you 15-20, because this is essentially just copying, okay? So 15-20 minutes if you just like do it. Make it neat. And then the back of this, I have it on two separate papers, but it's the back. You grab it on the handout table, is the Gizmo. And you're going to go to TAM Portal to access the Gizmo. It's called Cell Division. And you're going to be taking data on how many cells out of 100 cells are currently in interphase versus prophase, metaphase, anaphase, telophase, cytokinesis. So how many out of 100 are in each of those phases over 4 trials? Just use the instructions. Make sure you click record data, otherwise it won't give you the data. And then you're going to average it out. We have calculators in the student supplies cabinet, but you can also use your phone. Just don't be obnoxious with it. So find the averages, and we're going to use that data to inform us on how much time is spent by a cell in any of these. Because if we see that most of the cells are in interphase or prophase or metaphase at any given time, then cells are probably spending most of their time in that phase. So that's what we're investigating. That's activity B. And then the extension piece, which everyone's expected to do on the Gizmo, is kind of observing how rapidly the cells in this multiply. This is really setting you up for our next bit of this unit. Right now we're talking about regular typical cell division. We're going to start talking about abnormal cell division next, and why that might happen. So we're doing this, and your foldable. I would estimate all of this stuff all together should take you about an hour, maybe less if you're doing it and being focused. Remember, you're using color on this. You do not need to use color on your foldable. You got it.